This technology was used to modify the genomes of other species to produce desired products such as animals or plants. In 1952, Hersey and Chase made a breakthrough experiment that DNA is the fundamental source of our genetic code. In 1980, the first mouse was genetically modified, and in 1982, a giant mouse was produced by injecting growth hormones from a rat. This technology is used in order to take DNA from different organisms and combine them in order to create a new organism. It is also used in order to produce a useful or desirable characteristic in an organism. People with diabetes are lacking in insulin. This technology helped create insulin protein which can be injected into an individual with diabetes in order to help them survive. This technology has positively changed the direction of medicine both medically and therapeutically. Gene therapy is an important technique that uses the principle of genetic engineering to address diseases. In 2015, 45 clinical trials with gene therapy were approved and took place. Some trials treat conditions such as AIDS, cystic fibrosis, high cholesterol, and a variety of cancers. Farmers use a variety of genetically modified plants that contain herbicide and pesticide-resistant genes, thus allowing them to use chemicals without the worry of ruining their crops. In 1997, there were 17 genetically modified plants with herbicides in them, where in 2015, there are 97 of these plants in the U.S. An example of further research being done is the modifications that may occur directly in utero. Scientists believe that they may be able to terminate a majority of inheritable diseases. They also believe that they will be able to make physical changes in strength, height, and appearance, as well as make changes in intelligence and personality.
Genetic engineering is the technical process of inserting DNA into an organism with the purpose to add a characteristic. For example, human tissues are being grown in and on animals for transplantation if the human lost or is lacking it. GATC are the building blocks of DNA. Each species has its own sequences, making their DNA unique. The only reason genetic engineering is possible is because all Earth's organisms share this universal genetic code. Genetic engineering works by implanting DNA from one species with a desired trait into a completely different species so that it can express that gene as well. The recipient organism is referred to as the host. DNA implanted into the host can either be artificially made by copying DNA sequences, or it can be physically removed from the giver organism by radiation, drugs, and other techniques. Vectors act as vehicles to deliver the DNA from the giver organism into the host organism without harming its cells. There are multiple types of vectors. One example is a chromosome. Viruses are also used as vectors. The gene of interest is put into the virus, and the virus is injected into the host organism. And as viruses spread very quickly through cells, so does the gene of interest that they contain. The most common vector is called a plasmid. Plasmids naturally form in bacteria, so this method is usually used when bacteria is the host. A plasmid is a double loop of DNA separated from the nuclei in the cell. It is less risky and simpler to use plasmids because we are working only with some DNA as opposed to the entire genome in the nuclei. The plasmid requires 3 components in order to successfully function. Multiple cloning site. This is where the gene of interest is inserted and replicated many times. An origin of replication. This ensures that the plasmid will divide as the cell that it is in divides. A selectable marker. This is a gene in the plasmid that tracks the cell with plasmids inserted into them by killing other cells. For example, if our gene of interest is insulin, the insulin gene is inserted into the multiple cloning zone, and an example of a selectable marker is an antibiotic-resistant gene. This way, to identify the cells with the plasmid, the cells are exposed to an antibiotic. The cells that survive contain the plasmid, and the cells that die don't. The use of genetic engineering has benefited our world in many ways. In comparison to artificial selection, genetic engineering is much more precise as we have control over which gene changes are added, focusing on one gene at a time as opposed to many at once. For example, crops have been genetically modified to grow faster and tolerate environments where they couldn't survive before, such as drought, salty soil, and cold areas. This has allowed for more crops to grow in a shorter period of time. Genetic engineering has also enhanced nutrition in these crops as fatty acids have been altered, boosting protein levels. In addition, they are healthier and cheaper because tons of expensive chemical pesticides are no longer needed to be sprayed. Genetic engineering has also enhanced the pharmaceutical industry. The human growth hormone and insulin are very rare but very precious to our medical field. So transgenic organisms, most commonly bacteria, carry these genes to develop and multiply them as bacteria multiply very quickly. Although genetic engineering has improved many aspects of our world, it also brings many issues. Changing the natural state of an organism can unintentionally affect multiple of its characteristics, including metabolism, growth rate, and cause allergies. New allergies could also arise during the process if a desired trait is linked to a gene that causes allergic reactions. Insulin used to be grown in cows and pigs, but it would give humans allergic reactions, therefore other host animal options had to be explored. When GMOs, for example salmon, are released into the environment, they can be controlled and could harm and disrupt the balance of the natural ecosystem. They could outcompete natural species and interbreed and create undesirable offspring. From a religious point of view, genetic engineering could modify organisms so much to make them better than the creators, which will arise conflicts. Finally, as genetic engineering has not been around for a very long time, we don't know the long-term effects of it.